So today we're going to be talking about the ABO system, and this system refers to the molecules on the surface of red blood cells, and they're classified depending on what these molecules are into this ABO system. Now, as you can see here, I've drawn 4 red blood cells, and they each have different combinations of molecules on their cell surface. So going from left to right, we're going to work through these. Now we're going to start with phenotype. And as you can see from the legend here, this first red blood cell is type A. And this is because it only has the A molecule on its cell surface. Now, this is because of its genotype. Without getting into the exact genetic basis of the ABO system, which is a little bit complicated, we're just going to say that it's an example of codominance and leave it at that. So that means that this red blood cell here, that is type A, must have at least one of the A alleles because it's expressing it. Now that second allele that it has to have can either be the same or it can be different. And this is because in the ABO system, we have the O allele that essentially does nothing. It means that no molecule is expressed. So either of these could give rise to a type A red blood cell. And the final thing that we want to consider, which will become important later, is the type of antibody present. Now, as we know, antibodies in our body are produced to foreign molecules. With somebody who is a type A, they have the A molecule, but not the B molecule. Therefore, the B molecule will be considered foreign, just as the A molecule is considered self. So they will have anti-B antibodies. So now let's move on and do this for the other 3 red blood cells. This one here is type B, and this is because it has the B molecule on its cell surface. And similarly to type A, the genetic basis of this could be this genotype, or it could be this genotype. And again, this is because of the principle of codominance. Now, when we get to this red blood cell here, type AB, we see that it has both molecules on its cell surface. This is significant because it means that it must have this genotype. It can only have this genotype because it must express both of them. Now, just to fill in what I forgot before, the antibody present in type B will be anti-A antibody because the type B blood has the B antigen. It cannot produce antibodies against that. And when we consider this in the context of type AB, we will see that there are no antibodies present because AB blood has both A and B molecules. Thus, no antibodies will be produced to either of these. Now, the final example is this red blood cell here, and it is type O. It does not have any molecules on its cell surface, which means that it can't have A or B genes. It must have 2 O alleles. And again, this is the only genotype possible. Now, if an AB blood cell, or someone with an AB blood type, had no antibodies, it's evident that someone with type O blood will have anti-A and anti-B antibodies. And this is because both the A and the B molecule will be considered foreign. So now, if we just move down, we're going to consider what happens when we transfuse blood from one person into another. So let's consider where we have a type A red blood cell, or we're taking blood from a type A person and we're giving it to a recipient. If we give it to a type A recipient, then everything is really perfect. The bloods come from the same person that it's going into, in terms of the ABO system, so there's nothing wrong. If we give it to a type B recipient, as you can see above, the type B recipient is going to have anti-A antibodies, so that's not going to work. If we put it into a type AB recipient, again, as you can see above, the AB recipient does not have any antibodies, and that means it will work. So the final recipient type is type O, and this recipient has antibodies to both A and B, meaning that this person will have antibodies against the type A blood, so that transfusion will not work. And this is why we often refer to type O as the universal donor. Because their blood cells do not have any molecules on the cell surface that are antigenic, it doesn't matter who you give their blood to. It will always be accepted. And this is also why we refer to type AB as universal recipient. Because in a similar fashion, someone with type AB blood does not have any antibodies that will target ABO molecules. And this means we can give them any blood without worrying about causing a cross reaction. And that's the ABO blood system.